Steve, thank you very much for joining us. I know you're a super busy guy. Being Canadian Blood Services kind of a microbiology officer during a pandemic is not an easy thing to do. Steve, you've got a very interesting CV that I'm going to share a little bit of with people because, you know, it's really interesting because it's kind of convoluted, isn't it? Steve did a BA in poli-science. Then he jumped ship to do a Bachelor of Science in Biology, Cell and Developmental Science, did a Master of Science, and did a PhD in experimental medicine. All of his post-secondary school education was at UBC. Yay. Then you did a clinical microbiology fellowship at U of T. And during undergrad, you did a stint with Canadian Forces as a medic. That's, like I said, a bit of a convoluted CV and very interesting. You're a well-rounded gent. You are now associate professor at University of Alberta in Edmonton. You, I guess you're associated with pathology and lab medicine, is that correct?
Yeah.
And you are also associate director of microbiology with Canadian Blood Services, and that's where I know you from. I guess you joined in 2018, and we met really soon after you just started, I believe in Toronto at the Canadian Society of Transfusion Medicine. And just as a little advertisement, there is the annual meeting for Canadian Society of Transfusion Medicine next week, I believe, starting on the 26th. Thank you very much again for joining us, and I'll let you share screen now, please, and tell us what you can tell us.
Sure. Can you see these? I think you can.
Not yet. You're not in presenter mode as yet.
Okay. Can you see them now?
Perfect.
Okay, perfect. So I'll get going here. So my name's Steve Drews. I'm the microbiologist here at the CBS. I have a few conflicts of interest. I'm a paid consultant to Roche on arboviruses and emerging pathogens. I do use Roche products in my routine and surveillance work as well. And I've received a support from Abbott to support a CHR funded as well as Alberta Innovates supported seroprevalence study for SARS-CoV-2. So I have two main objectives. Just see if I can move my face out of the way here. I'm going to review the seroprevalence levels in Canada and emerging evidence on minimum antibody thresholds that are required to confer protection against COVID-19. And I'm going to discuss pretty briefly, for a variety of reasons, whether SARS-CoV-2 antibody tests are clinically useful. So we're now in the 6th wave of SARS-CoV-2. The current epidemiology assessments have really been made difficult by a lack of consistent wide-scale testing across the provinces. But it's evidence that we've are probably on the downturn of this 6th wave, and possibly troughing out on that. You can see here some of the data across the country is not available for a variety of reasons, including the lack of consistent testing. The good thing about the last Omicron wave is that the ICU or intensive care unit and ventilation rates are still low. And so although we've had this significant but not absolute a peak like we've seen in other peaks, in this case here, really folks have not been overwhelming the ICUs and they have not been overwhelming the ventilator capacity. The current peak has really been dominated by Omicron, and this again is from Health Canada, a database. I'd like to bring this up, showing and the key teaching point here is that there's been quite a diversity of strains across the pandemic, and that strain diversity makes interpreting the protective capacity of our humoral antibodies or our humoral immune system a little more difficult. And I'll show you a bit of data, not including Omicron, that's been published from my group, which has looked at some of the differences that we've seen in donors donor profiles in terms of their responses based on their potential past infection or their vaccine status. One of the key things that I present to other groups, and I often update the slide set, really is that the burden of hospitalization and deaths is still proportionally higher in individuals who've not received a vaccine. And I think it's key to not look at the raw data, but if we were to generate an odds ratio from this, you would see that really those rough back of the napkin calculations would really be balanced towards, if folks are not going to get vaccinated, they have a higher proportional risk of death or hospitalization. This is a very basic slide that I've had since probably the very start of the pandemic on how we develop immunity. And one of the caveats I'll give today is that I'm not really going to address the cell-mediated immunity at all. I'm really focusing on antibody-based immunity and potentially how some of that immunity might change, or at least immune responses might change across variants. I'm also going to think about or talk about, really trying to understand whether we can have a true correlative protection at this point. And so we can see from these cases here that, with vaccinating as well as natural infection, individuals are going to generate those antibody responses, but it's not going to be, they're not going to be homogeneous. And really, I think the nucleic acid vaccines that we've been largely exposed to, with the very high levels of protection or vaccination in the population, have been a great job in driving some of that neutralizing capacity up, at least when we use lab tests to look at that. I think when we think about serology testing and we think about antibodies, we have to be careful to kind of get a sense of that we're going to have tests that give us qualitative results. So it was an antibody present and to what marker or what SARS-CoV target. Some assays, such as the Roche anti-spike assay, or as well as the Abbott anti-spike assay, will give semi-quantitative values that are not necessarily yes or no, but maybe give a value. It gives some sense of neutralizing capacity to infection. Other assays will give quantitative or neutralizing results that can be related to a bioassay, like a print plaque reduction neutralization test. I think it's key because how do we measure? If I can go back here, we're really going to be thinking about, how does that neutralizing capacity work? We're really thinking about, in many cases, blocking that interaction of the spike protein on the virus with the ACE2 receptor or co-receptor activity during that invasion process. And so we've classically thought about blocking that ACE2 and spike interaction as a protective approach, and an approach that we've often thought about when thinking about convalescent plasma, for example, or monoclonal or polyclonal neutralizing antibodies. So I think here at this point, I think it's key from that fourth point there, or the second bullet point, is that there's currently no recognized public health or clinical indication for the use of these semi-quantitative or quantitative tests. And we're using neutralization assays, and in some cases semi-quantitative assays, to try to identify possible correlates of protection in epidemiologic and clinical studies through a variety of approaches. And I think we also can't fall into the trap of forgetting about cell-mediated immunity. But again, I'll focus on antibodies for a variety of reasons. So I used to be responsible for respiratory virus testing in the province of Alberta. And I built a career studying emerging pathogens and respiratory viruses. And I jumped to Canadian Blood Services thinking that I would never have to deal with respiratory pathogens again. And that was in 2018. In 2020, I was engaged as a microbiologist to support convalescent plasma studies by trying to find tests at that time that would allow us to qualify a convalescent plasma for clinical trials that were occurring in Canada. And historically, to put a perspective on this, at the time, there were not a lot of either commercial or lab developed serology assays for SARS-CoV. So I did a lot of work in those early months, building relationships and trying to think about which assays might be appropriate. Around the same time, I was starting to become engaged on early seroprevalence studies, and there's two in particular, CIHR-funded study, or an Alberta Innovates funded study that I was a PI on, as well as a second study that was through the COVID Immunity Task Force that Dr. Sheila O'Brien at Canadian Blood Services was heading and was testing a fairly large number of specimens. Both so I think we'll go over those numbers as we move through. So we previously discussed on this, but how would the concept of a neutralizing antibody work? And we're really thinking about that spike to ACE2 interaction primarily, and then really thinking about that receptor binding domain on S, mediating both the viral binding and fusion events, and simplistically suggesting that the neutralizing antibodies would block these binding events and fusion events. But I think when we started to do the work, we realized that not all antibodies were neutralizing, and that humoral immune responses to SARS-CoV-2 infection or vaccination could include both the binding antibodies and neutralizing antibodies. And I've broken them down into two broad groups at this point. This is a study that I was involved with that was with Anne-Claude Gingras out of Toronto. And essentially, you have a variety of a really messy graph here, but on the y-axis is an area curve of displacement at competition assay, which was considered a proxy assay for neutralization with S or spike. On the x-axis on the bottom, you have an area under the curve of direct binding curves for those assays. So as you're thinking about basically running these, you think about a lower area under the curve for that displacement potentially being more neutralizing, and then the area under the curve of the binding assay, as it increases, you would suggest possibly that there was more neutralizing capacity to the receptor binding domain. The black circles represent PRNT, or plaque reduction neutralization 50 results. So the bigger the result there is a bigger PRNT 90, and the darker color on those graphs, on those circles, suggests a greater neutralization capacity. The PRNT 50 really is a cell culture bioassay where you're trying to grow the virus, and you reduce plaques by using serum plasma that you provide from the patient or donor. So as you can see here, there's a wide variety of responses here. And these would have been collected very early on from individuals who we were using to validate our approaches. And essentially, these folks would have all had a self-declared NAT positive for a COVID infection, and had waited a period of time as well before presenting to donate for these specimens to use for validation. So you can see here that even on the upper left-hand side, there's some fairly big circles that are quite dark, some smaller ones too. So not all, so not all these antibodies are going to be neutralizing, and not all these antibodies are going to be binding either. And you're going to have some cases, folks you think should be generating fairly strong responses who are not. So we also had a fair number of folks who declared an infection and waited the time to generate a good humoral response, and we could not measure anything off those folks, not even binding antibodies. So we were getting a sense very early on in the spring of 2020 that the humoral responses to infection at the time, because again, this was before vaccine, were quite diverse. As we started to support the convalescent plasma project, we noticed that a lot of folks would come in, they would give a product, and we would qualify it. At the time, with the PRNT assay, we were qualifying plasma with a titer of 1 in 160, and we dropped that down after a while to 1 in 80. But individuals would come back, and we published this in Transfusion in 2021. If you look at the y-axis here, you can see the PRNT50 titer, and on the bottom here, this is the days post onset of signs and symptoms. What you can see here is a lot of folks really had crashing titers for PRNT50 results over about 3 to 4 months. And in many cases, then they would come back and not be able to qualify the plasma for donation for these trials. So this again was not something we expected, although it does make sense. It really is not probably a good idea to be constantly generating these massive titers. You should be letting other arms of the immune system protect you. But as you can see here, if you're trying to run a convalescent a plasma project where you're trying to get access to product, you're really going to have to keep recruiting people or get them in fairly early between plasma donations, or else you're going to be, in many cases, have folks who are just going to have crashing titers. There was a lot of other folks, up to 20, 30% who couldn't qualify on their first donation. Again, very much like the story I presented in the prior slide. So what does this mean? There were some early signals from the studies that they didn't see an effect, a beneficial effect to these convalescent plasma products. But there was a variety of issues with that. And one of them was, well, maybe they started these too late, or maybe they didn't titer them all the same way. We were providing product to the trials that was measured with PRNT. Other groups were providing product to the trials that were using other assays. There's an interesting signal I just highlighted here. They found that there were less harmful effects if the product, or there was a trend to less harmful effects if the product had a high titer via PRNT neutralization or proxy virus-like particle neutralization, or if there was a strong antibody-dependent cellular cytotoxicity assay profile. So there was a signal in here, and so it really got us to say, it looks like really when you're generating these products, you have to really think about that neutralization function, or that other function that's happening here that's supporting other arms of the immune system. And so again, I think it's key. There's more trials that are occurring. We're seeing a recent paper in New England Journal of Medicine, I think from a few weeks ago, suggesting that early application of these products might be successful. And there's also some evidence for signals, protection signals in individuals who have early product, as well as who are immunocompromised. So I think that, again, when we think about these responses, I think it's really important that we pull apart the different arms or the activities that these antibodies can be undertaking. When we're thinking about our protection levels and antibody responses in the population, I think we also have to think about what's going on in your local environment. This is a graph from a paper that we published recently in Microbiology Spectrum. And really, at the time, we were looking at collecting about 1,500 specimens a month between April 2020 and March 2021 from Canadian Blood Services donors. And so we used a variety of assays. We used anti-S and an anti-RBD assay from Anne Jean Gress's lab in Toronto. We also used one of her anti-nucleocapsid assays. These were IgG assays, and we used an Abbott IgG assay as well. And then we did what's considered, we found that there was really there was not a lot of discordance in many cases, or concordance, sorry, and that we would either use a composite reference standard of saying maybe 2 out of 3 or 2 out of 4 or 2 out of 5. Positives might score a positive. We also did something called latent class analysis, which my colleague Dave Fissman and I have been looking at probably for the last 15 years, and trying to model a gold standard where you don't really understand what the gold standard is. I think the key story here, regardless of the modeling, and what we presented is a seroprevalence percentage here in this blood donor group of 18,000 specimens, I think -2. And we have the confirmed cases in the population from PHAC in the gray, and you can see here that we're tracking fairly closely here with a little bit of a timeline, of course, that you would expect to see because you're measuring humoral responses to infection. I think it's key here, though, the key picture here that compared to other jurisdictions, going from April to March, so April 2020 to March 2021, we really didn't cross that 10% threshold in the overall population for antibody responses to SARS-CoV-2 infection, regardless of the marker when we corrected for these, which is important because I think when we think about looking at the population and understanding protection as well as what's going forward, I think we have to think about pre-vaccine era, we have to think about vaccine era, we have to think about the interaction as vaccine and infection, and then we have to think about what happens when you have significant breakthrough infections in a highly vaccine population, such as we're seeing now, and I'll show you some of that data later. We also, some of the problems that we had early on was that we saw waning signals, in particular for the Abbott nucleocapsid IgG assay. As you can see here at the bottom, it would start to tunnel out here after a few months. Now we didn't phase it here, but we could show in other work that we were starting to drop those titers over time. And nucleocapsid is not the only marker that wanes. And I'll show you some data in a bit for spike. But you can see here, regardless of the assay, there's a Sine assay here, and you can see both very similar drops in the orange and the green. So this one here and this one here, although the amplitude is slightly different. You can see here that the RBD and the spike signal retain themselves, but I'll show you some data later showing that those will wane as well. So interpreting these results, if you don't have a time point of past infection exposure or vaccination, it makes it difficult to interpret sometimes what these serology results mean at a global level. And there is no serology gold standard. So this is something that just is coming out in press, microbiology spectrum, and this is part of our group as well. I just wanted to impress on you here that over here are positives that we've tested by a variety of methods. You can see some will only agree with 1, 2, or 3 results, and some won't agree with anything. And these were scored as positives. Here are some negatives. You can see here that the assay specificities in general are a little bit better in agreement, but that's not always correct. But I think the concept here is that in many cases we were struggling, and I think we still struggle with what marker to use, and also, is it an issue of sensitivity in the assay, or is it something else like waning? So that's something, again, we have to think about when we look at these results in terms of population level data, as well as looking at whether you would look at an individual and say, well, it looks like you have evidence of an infection or a vaccination or not. The work by Sheila, and I supported that, was larger. This is a CITF supported, or a COVID Immunology Task Force supported project. And the scope here was bigger. So again, we sampled blood donor specimens from across the province, but the numbers here would vary. So instead of doing 18,000 a year, would go from 15,000 a month up to possibly 40,000 specimens that were plasma specimens that were tested. Originally on the Abbott system, that was a nucleocapsid IgG assay that I've kind of described before, but then on a Roche spike and a nucleocapsid. So let's go and look at the blue line first. So again, this is seroprevalence here, and you can see the month down here. And as you know, again, very similar. We started in April, essentially moving up to January, and very much like the CIHR data, prior to March, we were really sitting at 10% with a spike signal here. You can see that the nucleocapsid is a little bit lower, with again vaccination rates in Canada and the campaign to do that. And blood donors are highly compliant in terms of health issues. You can see here that by probably October or November, almost all of our blood donors had evidence of antibodies to spike. And this is a spike total assay that gives us a semi-quantitative result. So we're measuring IgM, IgA, and IgG here against the spike, but primarily against the RBD. And so this could be used also as a qualitative result, as you see here, but also as a semi-quantitative assay. The signal here, again, it suggested that we weren't getting a lot of infections naturally, or that there was some waning going on. You can see going into November of 2021 that we still, again, had fairly low levels of nucleocapsid signal here, suggesting that even though we suggest we'll probably see some waning, that we had a very low level of breakthrough infections in our population going into the winter of 2021, 2022. This by March had gone up to, in general, in our donor population, which represents individuals from across the country except for Quebec, about 30% positivity for that nucleocapsid signal. So you can see here, even though these high levels here are at least a high proportion of population of donors that were probably vaccinated, we still were getting a significant nucleocapsid signal suggesting breakthrough infections. So again, underlying key story here, even though you've got these antibodies that are probably doing a pretty good job, you're still getting this infection coming on here in your population. This can be broken down in a variety of other ways. You can look over time. You can see by region. For example, you can see in Alberta here how it slowly crept up from December to January. You can see younger folks were probably between the 17 to 24 age group, were probably out doing more social activities and becoming infected as well. So I mean, you do have that waning approach, but this offered an interesting perspective, because prior to this, we really did not have that much exposure to natural infection in the population. So it was easy to kind of pull these apart at the time. Once that cat or the horse is out of the barn or the cat's out of the bag for this kind of thing, it becomes more difficult to interpret. And it also becomes more difficult not to look at other aspects of the immune system. I think by March 2022, really, again, if you look at the spike result here, you see high levels of anti-S that have been adjusted. And over here, you can see that there is, again, fairly high levels of anti-nucleocapsid. And down here, you can see that the younger age group, almost at this point, around 44% of them, this blood donor group, were showing evidence of a nucleocapsid signal. So a fairly big Omicron wave impact on this donor population, even with what looks like to be a vaccination state, at least one a dose of a vaccine in that population. So what's the value then of looking at SARS-CoV-2 serology testing? From a blood operator perspective, from a transfusion perspective, we're probably considering qualifying convalescent plasma. It also provides us with the ability to do seroprevalence studies and surveillance. From a blood operator perspective, this has really moved us from, again, being focused, on supporting the production of blood products to supporting public health at the provincial and national levels, and engaging working groups with that data. And also thinking, okay, well, can you identify correlative protection? So far we've looked at neutralizing antibodies drop, and a big chunk of folks, some people, even though they've been infected, don't have evidence. Other folks will have evidence of what looks like a vaccination, but still have breakthrough infections. So what is a correlative protection? Simply, we're looking for a molecular biomarker to measure how much immunity is needed to fight an infection. And this is key for successful global immunization programs. So, we have correlative protection for things like hepatitis B. We can measure how much antibody they have to hep B surface antigen, and have a good sense that most people, if they were to have a blood and body fluid exposure, would be protected against that. I don't know, and I'm going to argue that I don't think that is the case with SARS-CoV-2. So this is another paper we put out from the CHR and Alberta Mandate's work. And really here, without going into too much depth, this is relatively small groups, but at the time we had the capacity to measure these folks. We pre-chose individuals based on what looked like their evidence of infection. based on binding antibody responses, and then took those specimens and ran them with viral-like particle neutralization assays for wild type, alpha, beta, gamma, and delta variants here. At the time, we didn't have Omicron, and we were still moving along with that. And then we had the ability to look at, because this group was collected, had collections done between January and March 2021, we asked the question at the time whether they had a vaccine or not and what the vaccine was. So we could pull apart, without too much granularity though, whether they had a vaccine or not. And so we also could pull apart whether they had an anti-N signal or not. So this group here would have been vaccinated and had an anti-N signal, so suggestive of a natural infection plus vaccine. This group here was vaccinated but had no anti-N, so this was probably the, like most folks, a naive group in terms of infection but had a vaccine. This group here was unvaccinated and had an anti-N, so suggesting that they had not been vaccinated but had been infected. And the other group had no vaccine and no anti-N, but did have binding antibodies, and those folks were not on this slide because they had no neutralizing capacity at all in this assay. So as you can see here, just briefly, individuals who were infected and had anti-N seem to have very broad neutralizing capacity, with the exception of possibly some drop here in beta. But you can see here that these folks in general, their medians, people had variable responses, and these are small numbers, but it shows you here that in most cases you can have something to neutralize if you've been vaccinated or if you still have that evidence of a natural infection. So the group that had no nucleocapsid here, again, didn't neutralize, but you can see here that there's still protection across the board. But this group that had an infection and had been vaccinated seemed to have a broader capacity. That I'm not arguing here that people go out and get infected. This group here still had very good responses, albeit there were some drops. And you can see that beta, again, is a classic story here, probably some escape here. Whereas in delta, we were really thinking about less of escape and more of a titer effect here, with higher titers possibly in the upper respiratory tract. So we also saw that anti-S rates increased dramatically, but as they increased, you could see some sort of a waning capacity. But this group here is actually, some of these folks were the same folks that we saw in that kind of really messy XY graph with circles that were big and little. But you can see here that we validated these folks against print 50s here, and then this Roche anti-S. And you can see that the cutoff here is actually, for the quantification, the qualitative cutoff's 50, but the quantification can go down under 1. You can see some people with fairly strong responses in the 300s. This is not uncommon then in the first wave to see people here. This assay they would have to have their specimen diluted 1 in 10 to get this value above, I think it's 250. So really, what looks like fairly strong responses. When we started to get into vaccine era, if you look on the y-axis here, if you think here, well, the highest were 450, 500. Here, we were seeing people up at 5,000, 20,000, some folks up at 100,000. We were running by, I think, halfway through the vaccine program, we started to do a 1 in 400 dilution to try to get titers that were measurable for the spike assay. So again, in the presence of vaccine versus natural infection, you can expect to see fairly massive increases in these anti-spike results. Interestingly, you can also see that those will start to wane over time. And they'll wane differently in different age groups. The first waning signals that we saw were really in this group of 70 plus donors, but you can see that across the board here. Now, does that mean they're not protected? No, I'm not arguing that, but I think if you're trying to measure these signals and look for protection that way, a correlative protection that way, or if you're going to try to do convalescent plasma programs going forward, you have to take that into account in your population. Again, the same story, just to show that your anti-S signals will wane over time as well. And so that nucleocapsid is not the only target when we're looking at antibody responses that will wane. So is there a correlative immunity? There probably is some evidence that vaccine effectiveness studies that we can probably pull some of that out of there, but I don't think it's as hard a cutoff as, let's say, going with some other target like what we know from hepatitis B work. Again, you could see here that they had a vaccine effectiveness of 80% against symptomatic infection, which was mostly at the time alpha variant. I think that's key. They could pull it out as 264 binding antibody units for anti-spike, or they could use a neutralization assay as well down here at 247. So some of the issues here are standardizing. We can standardize pretty well for VAUs now. This will really neutralizing capacity down here is really going to depend on the assay you use. So there's probably some evidence that there's some protection, at least for an early period of time after vaccine, where you could use these to measure that, but I don't think it's consistent. And I think the story that I've told you already, what I'm trying to paint here is that there's a lot of other confounders out there that will make some of this work difficult to try to apply in a larger scale and in a clinical setting, or even to support public health activities that are focused on finding a correlative protection. So maybe I'm arguing is that both neutralizing and binding antibodies are correlative protection, and I think that the story is a little more complicated than we first thought when we walked into it in April 2020. So we probably have to think about that correlative protection would have to involve both neutralizing and binding antibody responses. So this is kind of where I ask folks what they think is the most appropriate answer here. So choose the most appropriate answer regarding your understanding of humoral immunity. Do you think the presence of antibodies to SARS-CoV-2 suggests that these antibodies are neutralizing? Do you think that antibodies to SARS-CoV-2 spike protein in a donor mean that these antibodies are neutralizing? So the difference between antibodies here and spike protein antibodies. Do you think that antibodies to SARS-CoV-2 may or may not represent a population of neutralizing antibodies to SARS-CoV-2? So again, it could be either spike or nucleic capsid, but I don't, it may or may not. And then the D one is where I'm kind of sitting at still. I think I'm hedging my bet. There's just too much variability with these immune responses to describe here. So there's no tool here. You're not going to be asked to vote. You just can put your answer in the chat, and maybe we'll get some ideas what folks think.
What's the question?
We'll do the questions afterwards.
Okay. Yeah.
So we have some C's here, I think. Okay, I think we'll move on here. So my argument is that the roles of antibodies to SARS-CoV-2 are complex and are still being studied and understood. I think that we have a sense that they're polyfunctional. There's probably antibodies that are involved with neutralization, and there's probably antibodies involved with cell-dependent, cell-mediated cytotoxicity, or even cell-dependent or antibody-dependent cell phagocytosis. I think the ADCC and ADCP components here are probably still being understood. And I think from our perspective, there are still some activities that we can be doing with serology assays that will be beneficial to public health and maybe blood operators in general. But should you be testing folks for antibodies? and what are the general perspectives on what the profiles might look like. And this is from the CDC. So a person who's been vaccinated, or a person who's never been vaccinated, they could have a marker or antibodies against nucleocapsid spike or RBD, and that might, would again be suggestive of prior infection, although you can have some false positives, and that might be creating some noise in some of the assays. I don't think testing people who've never been vaccinated for these signals is a good idea. This is something that's not really gained traction in clinical labs or public health labs. And again, you might see waning responses. We also know that some folks just didn't generate any antibody responses that were measurable at the start using some of our binding assays. Other folks wouldn't have generated a strong response using neutralizing assays either. And so in other cases, testing people for nucleocapsid, they might have a past infection, but in some cases there could also be false positives depending on how you set your thresholds and what is the gold standard. And I think I've touched on some of that as well. So there's probably some role here. There's a document, second documents from Alberta Health Services. So antibody testing really is not a frontline test in clinical public health labs. And there are some scenarios where individuals, it might be beneficial to help the clinicians work through what's going on in some fairly complicated clinical pictures. But given that these antibodies might persist for months, possibly years, as well as waning events, there's some major caveats here. Now, that said, we still look at in populations prior to massive vaccine and then Omicron, we were still measuring effect using nucleocapsid target, for example. So if we're thinking from the clinical perspective, don't replace virologic testing with serology testing, and don't use serology testing for measuring immunity following infection. Don't use serology to assess for the need of vaccination in an unvaccinated individual. And we need to don't use serology testing to establish quarantine rules either. So I think from my perspective, what I've tried to suggest to you is there's a diversity of these antibody responses in individuals who've been infected with SARS-CoV-2. Neutralizing and non-neutralizing antibodies will probably play a beneficial role in immunity. We can use some of these assays to support our surveillance and therapeutics programs, but they have limited clinical roles. And that the picture in Canada was really a very low seroprevalence rates early in the first wave of the pandemic. I think that with vaccination in Omicron, that these rates are going to continue to rise. Well, I think we'll see the spike maintain itself very highly above 95% for a while. But the data I've shown you for the nucleocapsid, I think, has suggested that will at least marginally creep up, although we might start to see that kind of stabilize depending on how much Omicron activity there is, as well as waning nucleocapsid signal. And I think we're still struggling to understand correlates of immunity to SARS-CoV-2. So this is another one here. So choose the most appropriate response. So we're confident that humoral immunity to SARS-CoV-2 is long-lasting and protective. We think that humoral immunity might not be completely protective, but is a good marker of past infection with SARS-CoV-2. We're still developing knowledge on this, or there's just too much variability, and with the variants, it's going to make it even more unclear. So I think of a variety of B and C here. So there's a lot of people. I presented some of our work, and some work is from the literature. This work that we did was supported by CHR and Alberta Innovates. We did a lot of stuff with the Canadian Task Force. We had a lot of collaborators across the country as well as in the US. And we continue to work with a group of folks out of the U of T, or U of A, and Canadian Blood Services to understand what's going on here with these responses. And I think that's it. Oh, and then, would you hedge your bet or not? I think the group here is I've seen some comments, but it would be interesting. I'm going to stop sharing here and just go back to my picture.
Steve, thanks a ton for that really interesting talk. A lot of questions. Dieter, do you still have a question that you'd like to pose to Steve?
Yeah, I would like to do it. So thanks first for the really nice paper or for the presentation. So I have a question. So I'm wondering, how compares the antibody waning kinetics for the S protein with any of the other vaccines, like diphtheria, hepatitis, and so on? I mean, antibodies are, for me, as a layperson, antibody, right? Are they wane all the same way? Or are some of the antibodies last forever, essentially?
So, I mean, I'll frame it in a, so from within the SARS-CoV kind of data we have, it really depends on the assay that you're using as well as the target. So as within SARS-CoV-2, we saw signals wane more with some commercial products than with on lab developed assays. And then we also saw that the nucleocapsid signal generally waned earlier than the spike. The classic, coming from a public health background, I mean, the classic correlative protection would be having a strong neutralizing or binding response to Hep B surface antigen. So having a strong anti-HBs over time, we would often test for that and look to see if it was above, I think it's 10 international units per mil, and then establish that that individual was protected if they were in a blood or body fluid exposure, with the understanding that some people would be underneath that but have a strong amnestic response that would bounce that back. In those cases, individuals who were vaccinated with Hep B would have screaming high titers, and then that would come down gradually. So just with those two examples, I think it really depends on the assay you're using, the stability of the humoral immune response, as well as what components out of that are really protective. And then does it really matter in some cases, because your cell-mediated immunity will come back and kick it in. The other thing I didn't address today was...
But I mean, the question was really, let's say a tetanus antibody also, or the tetanus antibody, or whatever vaccination you had, right? Yeah. So, I mean, is it the same time zone? I mean, a couple of months, or some last for years, and looks like SARS lasts only for a few months.
It depends on the individual.
I mean, on average. I mean,
yeah. So some will last forever, like very long times, and in other cases, we might see them drop. And in some cases, it will really rely or depend on the vaccine that's provided, as well as the individual's baseline immune capacity. But yes, some like, I don't think with SARS-CoV-2 that we're going to have individuals with this measurable protective anti-S that would go out for months, and then we can say, okay, that's going to be stable. I think other targets that we use for protection, like for measles or for Hep B, those are different immunological scenarios that I don't think that we can say, okay, this is a protective level, we're going to go to public health and say we can set this here, and if we have a vaccine at that level for this, it's going to, we can say don't vaccinate these populations at a certain level. I don't know if I'm answering your question, but I think it's.
I mean, I was just wondering. I mean, it doesn't make sense to me to vaccinate every 3-4 months. I mean, then it's not a vaccine; it's a drug essentially.
Yeah,
And so then something is not working, to my opinion, right? I mean, with the vaccination approach.
So I think what I look down here, and I used to be in Wolf Jeffries' class like about 25 years ago. Anyways, but one of the things and no one asked me is I really just measured stuff in the blood. What we did, and we did in another paper, was look at mucosal antibodies in saliva, and I supported that work. It was done at Anna-Claude Gingras's group, and I think you probably to have a sterilizing protection against infection versus hospitalization, I think you need to have that mucosal protection. So good saliva neutralizing capacity, or at least the evidence of that. And do you have some B cells that are homed up in this individual's upper mucosa, and is that due to a natural infection? I think what we were missing is like a variety of things, but one is that the vaccines that we have right now are intramuscular and really focused on that non-sterilizing response. And so I think if you could get upper tract nebulized vaccine in your population, you might be able to get some protection in there. I think the data that I've shown you now is all over the place, because with these breakthrough infections of Omicron, we probably have folks now with some fairly strong mucosal responses, and there are studies that are starting now to look at that in saliva. And actually the kits are being sent out, I think, this week. So I think that's the other gap. The other thing from the WHO conversations that are happening is that I agree, we can't keep boosting people, or not even boosting. We're not boosting them. We're just giving them the same dose over and over and over again. Is at some point, do we think about changing our approach to application of vaccines? And then what kind of vaccine does it look like? And should there really just be a pan-sarbecovirus vaccine out there? And do we chase after variants? So I think we're missing a whole bunch of stuff when we think about these correlates of protection and what we should be measuring.
Oh yeah, thanks a lot.
Wilf, you wrote a chapter in the chat. Would you like to unmute yourself?
Okay, thanks for the lovely talk, Steve. And I do have a question, and that is, my understanding, at least reading the literature, is that there's multiple units being used to describe antibody levels, quantities. And I've looked into this quite significantly. And I'm, for the most part, the WHO is advising everyone to compare antibody levels to a single patient, the antisera from a single patient. But now that antisera is temporarily unavailable for distribution. So I'm just wondering why the field hasn't gone towards using a quantitative measurement like nanograms per mil, or something that's not going to be affected by these international units. So
that's a tough one. I mean, I think we didn't have, I mean, at the start, we didn't have a gold standard. Everyone was using different assays. Everyone was using different chemistries and different targets. Getting enough material was tough. There is some work being done by folks who moved out of the WHO setting into California, I think it is, who are trying to build a broader selection of standards. I think my gut feeling is there were a variety of operational and study issues that forced groups not to have a standard approach. Some of that for the semi-quant assays can be brought down. So the commercial assays, if I'm comparing an Abbott anti-SIgG semi-quant against a Roche, I can do some calculations post facto to get them into what's called a BAU, binding arbitrary units per mil, which kind of standardizes some of the work. The neutralization assays for the virus-like particles were not all consistently done across the board. So some of either the cell lines they were using or the constructs were different, and the reporters constructs were different. From the plaque reduction neutralization perspective, it was really lab to lab, and then also really dependent on the cell line you were using and whether it had been recombinated or not. Omicron seems tougher to grow as well in culture and have it standardized against everything. So I think just because it happened so quickly and then there are just so many moving parts, I think that's been a problem. And I agree, I think it has to be more standardized, but for a variety of reasons, and they can be from science all the way out to politics. I don't think there was a consistency there. I do think we need to have a less biased and arbitrary control comparator, but really that's one of the issues now is how do you compare across tests or papers? I probably didn't answer your question.
No, I think you did. You confirmed my doubts about the quantitation, and I think it's a major problem, especially in the future when people do come to a standardization, a standard methodology, and then we try and go back into the literature to understand, papers that didn't use those standards. I don't know how we bootstrap ourselves back to that literature, and it's going to be a real problem, I think. So.
It's tough. I don't, again, like, I agree with you. I think you can look for general trends, and some of the problems is interpreting what happened and then trying to pull apart the chemistry of that experiment to suggest whether or not you can actually apply it one-to-one or just look at the general trend. So when we're doing some of the stuff, we're saying, well, when I look at this assay versus that assay, so I look at a virus like particle assay versus a print, I don't see the same sort of minute changes that I can measure when find a p-value. I compare multiple against papers, and I can say, well, maybe there's some ordinal differences between my ability to neutralize a wild type versus a variant, which were the variants. But yeah, it's very difficult to try to make some common value and say, okay, this is a protective level.
Thank you.
Riney had a comment in the chat regarding Moderna vaccine booster given at lower doses and Pfizer full doses, and then some further discussion regarding that. I don't know if you wanted to articulate that, Riney.
It was just a comment because he said that the vaccine is in theory not a booster. It's like we're giving the same dose, but Moderna is actually a lower dose when given. So that's actually a booster, whereas Pfizer is a full. It was just a comment to what he said before.
I see.
Yeah, it's a lower dose, and I guess scientifically it's a booster, but theoretically we're not doing the classic, we're going to prime you and then boost you with lower. I mean, I think they are, but they're finding another vaccine, and then but that's not truly a booster from a public health rollout perspective. That's just a vaccine with a lower dose that you're using as a booster. I think it's going to be more difficult when you generate a public health response to say, what's my primary dose, and then what I'm going to do after 3 or 4 fairly high doses, and then I guess the question, I don't know, is do you drive folks to energy at some point?
All right. And Cedric, you just reiterated what Riney just said. So I think that unless you have something else to add, I'll just skip to Peter Grin's comment. Peter, did you have something else that you'd like to add further to what you put into the chat?
No, just that. Sorry, I'm losing my voice. So if you don't mind reading it, that would be great. Thank you.
Peter imagines that standardization is also difficult due to emergence of new variants. Would that be true, Steve? For instance, binding of the same antibodies to spike from alpha would likely vary substantially from binding to spike from Omicron with over 30 mutants.
Yeah, so we're actually crunching some data right now where we're looking at an Abbott assay, the semi-quant, and again, we're comparing that to print 50 results. And again, you can see this drop, and there's an ordinal difference. I won't get into, but you do see these drops. And so to say I'm going to use a semi-quant assay to try to show that I can get neutralization against a print assay, you're probably going to the lowest effective neutralization, or the strain that gives you the lowest effective print neutralization, and then above a specific cutoff, and then going to its top 95% confidence interval would probably say, okay, I can get it up there. But I agree with you. they've generated this off of wild type generally, and then or these targets are all proprietary, but you assume it's off wild type or fairly early on, and then you get this variable response that drops for things like beta and versus delta, for example, and then Omicron you expect it to drop too. There was a comment here too. I'll just go back. So I think Cedric asked what's going on with different jabs. There are studies out there. Shelley Bolden was doing one about U of T was doing one about getting a non-mRNA and then getting an mRNA. It's thought that some of these, so depending on the vaccine you get, you might be broadening the response as well. So I mean, really, there's probably not a huge amount of difference with the RNA vaccines, but if you move into vaccines such as the Novavax or Medicago vaccines with different constructs, you're probably going to get some difference in some of those responses humorally, or maybe through a vaccine efficacy study. But again, that's all over the place.
I had a question following up on Dieter's earlier question. Really, Dieter's got his hand up again, but I'm gonna interrupt you, Dieter, for 1 second. I was curious if there was something interesting about how SARS-CoV-2 might be interacting with the immune system, and I wonder if the natural serology might be different than for HCoV-229E, for instance, another very prevalent coronavirus.
Yeah, so I mean, some of the provinces like Alberta used to do significant amount of respiratory virus testing on stuff that was flu negative, and you used to see these peaks of seasonal coronavirus activity out there. There was some discussion early on whether some of the signals that we were seeing serologically in populations were due to cross-reactivity with other seasonal beta coronaviruses. We haven't really I haven't pulled that apart. I know that folks are trying to look at some of those interactions, but some of these folks probably still have some stable, not everyone, but some will probably still have some stable serology against seasonal coronaviruses. The other thing,
I just mean, are the kinetics of development of antibodies differing? And as an indicator that SARS-CoV-2 may be interacting with the immune system differently. And that's why we're seeing some really different sicknesses.
You know, I think, you know, I haven't thought about that. I'm just going to say that right now. But I agree that I think a lot of the sickness that we're seeing is probably due to a variety of other things, including autoantibodies and possibly, you know, antibodies against cytokines as well as adhesion or I'd say receptors. But there's also potentially there were some early signals on whether there was some protection from blood type as well, although we never pulled that out of the data that we had, and I collected blood type data early on, so it might be. I don't know. I haven't thought about that.
So sorry, Dieter, I cut you off.
No problem. I have another antibody kinetics related question.
Sure.
So I remember there was a Cell paper not that long ago, and they compared antibodies against RBD and against the internal domain. Whereas, you know, the RBD is supposedly neutralizing and the internal domain was potentially enhancing. And if I remember right, the enhancing antibodies seem to be more stable. So they waned less than the neutralizing antibodies, because after a few months, they found more enhancing antibodies than neutralizing ones. So is that not dangerous if you vaccinate all the time? Then at the end of the day, you end up with much more enhancing antibodies than neutralizing antibodies?
You know, I guess the answer I'm going to say is that there's a lot of data out there that doesn't make sense. At the end of the day, though, I'm not sure that some of that work really applies broadly when we think about vaccination campaigns. I think it's.
I mean, it was a paper.
Yeah.
I mean,
Yeah. I've also seen data out there suggesting that if your titers are too high, there could be a possible effect in some animal models or biochemical models. Again, I think it's something that you can study, but I don't think at this point it's something that says we shouldn't be vaccinating so frequently.
No, maybe we should study it, I would say.
Yeah.
Okay, thanks.
Steve, thank you so much for an interesting talk. A lot of discussion followed, but indicative of
problem.
that good talk. Thanks so much. And
Thanks.
everybody, thanks very much for joining us.
Take care.
Best wishes all.